to the Single Cell Spatial Analysis Program Seminar Series. For those of you who don't know me, I'm Justin Colstein. I'm faculty in the School of Public Health and one of the PIs of this program. SCSAP, as we call ourselves, this is supported by the University of Michigan Biosciences Initiative and is focused on building a community of researchers in the area of a high-throughput single cell and spatial analyses. To learn more about our program and join our membership, check out the link, which will be posted in the chat shortly. And then in this context, we are very lucky to have our special guest today, Dr. George Emanual. So Dr. Emanual is the scientific co-founder and senior director of technology and partnerships at Vizgen, and he's a real leader in this field. He spent the past decade developing high-throughput spatial profiling technologies, including extending the Merfish technology to profile 10,000 genes in a single sample. Pretty cool stuff. And so George received dual undergraduate degrees from the University of Colorado and then a PhD in biophysics from Harvard. And so today he'll be telling us about Vizgen's insight to spatial genomics platforms, and then make sure to stay tuned until the end for a special announcement of an upcoming pilot grant program featuring Merscope technology. And with that, I'll turn it over to you, George. Thanks so much for joining us today.
Great. Thanks for the introduction. I'm excited to be here to introduce the Merscope platform to your group. But it's, for me in particular, it's been very exciting to see this technology come all the way from just an idea back 10 or even 12 years ago within Xiaowei Zhuang's lab to demonstration in 2015 and the first Merfish publication to now a platform that the research community can more broadly use to investigate the spatial organization of their tissues. In particular, if we look back into the history of genomics technologies, if you think about where we are now in terms of our understanding of biology, we wouldn't be there without these advances in genomics technologies. That's all the way from bulk sequencing, where we're able to identify the transcriptome and the genome, understand what are the genes that are there within a cell, and then which ones are being expressed across different tissues. But as everyone here likely knows through the other talks through this seminar, single cell genomics is a recent advance in genomics technology, where maybe 5-10 years ago, instead of looking across a whole tissue, now we can dissociate the tissue into individual cells and characterize the transcriptome or the genes that are being expressed within each of those cells. So with that, you're able to start to characterize whether the different transcriptomic profiles that you see within your sample. But to really understand what those different cell types are doing, it's very useful to understand the spatial context as well, because cells aren't working within isolated droplets, but rather they're working in an intricate spatial organization. So being able to map hundreds of genes, which is enough to very finely differentiate the different cells that you see across the tissue, but all while maintaining that spatial context, gives you an additional layer of insight into biological tissues, where you can just take a tissue slice, perform a Merfish measurement, and map out exactly where each of these different cells that you've seen previously in single cell sequencing technologies is located. So now you can start to understand what are the different cell-cell interactions and such across the tissue. So it gives the spatial view into what was previously, prior to these types of single cell spatial transcriptomics platforms, only available within dissociated parts, where although maybe some spatial technologies existed, they didn't have the single cell resolution that would be required to map these fundamental units of biology back to their spatial context within the tissue. So what I'll be discussing today is first Merfish technology, how we're able to achieve this very highly multiplex spatial transcriptomics measurement with Merscope. From there, I'll describe how within Vizgen we've brought the technology from research lab-based technology, where it was invented within Xiaowei's lab, to now something enabled by the Merscope platform that allows a rapidly growing number of different research labs to start running Merscope measurements, Merfish measurements on their own samples, gain their own biological insight. And then finally, I'll discuss some of the exciting features that we've added onto the Merscope platform since launching it earlier this year, in January of this year, in fact, including protein staining capabilities and FFPE tissue compatibility that open up a lot of different avenues for Merfish technology and the Merscope platform. So at the end of the talk, I'll discuss some of the different applications we've seen with Merfish and how those different proteomic or with RNA multi-omic approaches along with the FFPE compatibility open up those additional avenues and additional application areas. But Merscope, as I alluded to, is a combination between Merfish technology and this easy to use platform solution that we've developed within Vizgen. With that, it facilitates the whole workflow all the way from custom gene panel design, where a Merscope user is able to pick exactly which genes they'd like to target within their measurement and measure that panel of genes that is most relevant to the biology that you're looking at across that tissue. With the Merscope platform, we also have reagents and consumables that facilitate sample prep and running a sample on the instrument. The centerpiece of the workflow is the Merscope instrument that you see here that I'll also be discussing later on, but that takes your sample along with the reagent cartridge, performs this highly multiplexed spatial transcriptomics measurement across that sample, and provides the processed results, the list of transcripts, the cell segmentation results, the cell by gene matrix, and such as output files for your downstream biological analysis. So you can focus on the biology rather than focusing on the details of how to get the technology to work. And additionally, we have analysis tools, in particular visualization software that lets you interactively look at the outputs coming from the measurement. So here with the Merscope platform, you're able to measure hundreds of genes within a single instrument run, currently up to 500 genes, entirely custom picked by you, but we're very soon expanding that to 1,000 gene custom panel. It's highly sensitive and accurate based on Merfish technology that I'll be describing very shortly. You can image up to a square centimeter of tissue. So when it comes to mouse tissue, that's almost 2 full coronal slices. When it comes to human tissues, of course, in many cases, for example, a punch biopsy, you can make a tumor arrays and look at multiple samples within the same measurement, but you can position tissue as you'd like within that 1 square centimeter of imageable, of selectable area. It's all inclusive, so you don't have to first run the Merscope platform and then run a sequencer. Rather, you get the data that you need out of the platform. And then with the instrument control software and the gene panel design application, we always aim to make it a user-friendly platform. With this, since shipping it in summer of 2021, officially launching in January of this year, we see a rapidly growing number of users successfully applying the platform to their different biological interest areas, a lot of neuroscience and oncology, but it's very flexible as a genomics tool. You can apply it to whichever biological system might have interesting spatial organization, which when you look at these multicellular biological systems, that's nearly all of them have some spatial arrangement that informs the function of the biological tissue. So first, for the discussion today, I want to start by introducing Merfish technology, which is what Merscope platform is based on, because once you understand the technology, then it gives a lot more intuition for how it might be applied to different research questions. But Merfish is an acronym. It stands for Multiplexed Error-Robust Fluorescence in Situ Hybridization. And as the name implies, it's building upon technologies that came before it. In particular, in this case, fluorescence in situ hybridization, or FISH. And it builds upon that by greatly increasing the multiplexing capacity. So rather than just measuring a few genes within a given FISH measurement, now you can measure hundreds, approaching thousands of genes within that single measurement. So the technology was originally invented and published back in 2015 from Xiaowei Zhuang's lab. Since then, there's been a number of technical improvements from within the lab and biological demonstrations, but that got us to the point where in 2019, we were able to get Vizgen started. Since getting Vizgen started, we've been able to facilitate adoption of Merfish through the Merscope platform with a rapidly growing number of institutions. But from the technology, from the way the technology works, there's a couple of technical advantages, all the way from very high cell throughput to very high sensitivity that as I go through the description, I can describe in more detail. But the starting point, since Merfish is built upon fluorescence in situ hybridization, I want to first describe how that process works. In particular, it builds upon single molecule fluorescence in situ hybridization. In that single molecule FISH measurement, the researcher designs fluorescently labeled oligos that bind specifically to a target transcript. So once these fluorescent oligos are hybridized against that sample and they're bound to only that target transcript, then when you look at this sample under a fluorescence microscope, then each copy of that transcript within that sample shows up as a single fluorescent puncta. So each one shows up as a diffraction limited spot. So then by counting the number of spots, you're able to very accurately characterize the expression level of that transcript with single molecule resolution. But since its imaging technology can easily image across very large areas of the tissue and maintain that single molecule resolution across that whole tissue, so then you can start to see which cells within the tissue might highly express that transcript, or even within a cell, do those transcripts co-localize with the ER or the mitochondria or with the nucleus to even get insight into the intracellular organization of the expression for that transcript. But as with a lot of traditional fluorescence technologies, single molecule FISH was traditionally limited by the number of distinct colors that you're able to image simultaneously. So here you see one color fluorescence image. Each spot corresponds with one copy of that transcript. You might be able to fit 3 or sometimes 5 or up to 7 colors within a single imaging round. But when you're trying to characterize hundreds of different cell types across the brain, that's not enough colors to really understand what's happening. So Merfish builds upon this concept of single molecule FISH by, rather than individually measuring each transcript, instead we assign each transcript a binary barcode. And then through sequential rounds of hybridization, we're able to identify where transcripts with each of those barcodes is located across the sample. And we'll from that perform a very highly multiplexed measurement, bump up the number of genes you're able to measure from 7 genes up to 500, or as we've demonstrated from the academic side, up more than 10,000 genes within the same sample. So the first step in that process, starting from an entirely custom gene panel, is to assign each of those unique genes across the measurement a unique binary barcode. So this is where the MER part of Merfish comes in, so multiplexed error robust. So these barcodes that we assign to each gene do have certain error robustness properties, where if there are mistakes that happen during the readout process that I'll explain over the course of the following slides, if mistakes happen during that process, we still have mechanisms to accurately identify the correct gene. So there's error robustness built into this process. But once each of the genes is assigned a unique binary barcode, then we stain the sample in such a way that we're able to read out that binary barcode using sequential rounds of single molecule FISH measurement. In particular, during that measurement, anytime there's a 1 in the barcode, we stain the sample in such a way that it shows up as a fluorescent spot. Anytime there's a 0, then that copy of that transcript remains dark. So for example, in the first round of imaging, we stain the sample so that any gene with a 1 in the first position becomes fluorescent, and you see this individual fluorescent puncta. So here, in this cartoon example, it's this blue gene has a 1 in the first bit position, and also the pinkish orangish transcript towards the top. So those have fluorescent dyes that will bind to them based on the encoding strategy. So you can image this fluorescent spot. Anywhere there's a transcript that contains a 0 remains dark. So then you save this image of the pattern of the fluorescent puncta across the sample. You extinguish the signal from the first bit and then stain for the second bit in the barcode. Now all of the genes that were assigned a barcode with 1 in the second bit position show up as fluorescent spots, because with the encoding scheme, the fluorescent probes will bind to those transcripts while all of the other transcripts remain dark. So again, we image this pattern of single molecule FISH spots, extinguish and restain for the next round of imaging, where it's a new pattern of single molecule FISH spots corresponding with the genes that have 1 in the third bit position. So once we go through this process of imaging all of the bits across the binary barcode, then we can look at those image stacks, look at where there's a bright spot and where there's darkness, in order to decode the identity of the barcode at each position across the sample. So for example, this one in the top left of the cell, you see a bright spot, and then in the last round, you have another bright spot. So that corresponds with a unique binary barcode. And since we know which gene was assigned to each of the barcodes, that gives us the gene identity for that position within the sample. So this enables us to greatly increase the multiplexing capacity of single molecule fish to hundreds or thousands of genes that can be measured simultaneously within the same sample. So then with that information, you can start to characterize how transcription is varying across the sample at a level of gene multiplexing that's useful for understanding the variety of different biological processes that could be happening with that sample. So in this way, Merfish uses the power of combinatorial labeling, sequential imaging, and error-robust barcoding to achieve this very high multiplexing, very high resolution spatial genomics measurement. So for example, if we look at a brain slice, by tiling multiple images across the area of the tissue, we get this transcriptomic information across the whole tissue, but also across that whole tissue, we have the single molecule resolution. So we can zoom into any region of this measurement and see exactly where each copy of each transcript is being detected. So with that, combined with cell segmentation, you can characterize how many copies of each gene you find within each of these cells to really finely characterize cell type. From the way that the technology works, there's a few technical advantages that I'd like to elaborate on briefly. So first, since it's imaging to capture additional areas of the tissue or additional cells, all you have to do is tile the XY stage across additional areas of the tissue. So with this, currently with Merscope, we can image up to a square centimeter of tissue. And within that square centimeter of tissue, there can already be several hundreds of thousands of cells that are being detected. Next, since it builds upon FISH, and FISH has been a standard procedure within the biological research community for decades now, it's been applied to a wide variety of different biological systems, anywhere from human to Drosophila to zebrafish, or even zebrafinch. And because of that, it's fairly straightforward to apply Merfish to new tissue types. So Merfish, by building upon fluorescence in situ hybridization, maintains a lot of that sample type flexibility. Next, Merfish is very sensitive. Since we're directly staining and imaging the molecules in situ, there's no need to extract the molecules out of the tissue or perform molecular biology to amplify those molecules. Instead, it's all directly detected in situ. Since each of those different steps of processing the nucleic acids, extracting them and amplifying them could result in loss of some of the transcripts, with this direct in situ detection, Merfish is able to achieve very high sensitivity. So we detect a very large fraction of the transcripts that are expressed for each of the targeted genes. This is particularly important with single cell level transcriptomics, because with single cell transcriptomics, for a lot of very functionally relevant genes, they're not all that many copies within that cell to begin with. So we want to be able to detect as many copies of them as possible. Next, with resolution, as we saw in the earlier video, you get this transcriptomic information across a variety of biologically relevant length scales, from the whole tissue level all the way down to the individual cell and even single molecule level. So you can look at how cellular neighborhoods are working, how cell-cell communication might be working, and also within the cell, what is the co-localization of those transcripts with different cellular components. And finally, with this binary encoding scheme, it gives you flexible multiplexing capacity. By just adjusting the length of the barcode, we can adjust the multiplexing capacity of the measurement. But probably most importantly, for any new technology, it has to work well when compared, or it has to be consistent with technologies that came before it. So when we compare Merfish measurements with Merscope to existing well-established technologies like bulk sequencing, we get very strong correlation between the copy number detected with Merfish and the abundance measured through a bulk sequencing measurement across very wide dynamic ranges. So here we can see nearly over this more than 4 orders of magnitude of gene expression measured by bulk sequencing, we get linear correlation with Merfish. So as we'll discuss in some of the examples coming up in the later sections, beyond just this good correspondence correspondence with bulk sequencing. We also get very good correspondence with single cell sequencing as well. But when we compare this to spatial technologies that came before it, for example, spot-based, array-based technologies, there's dramatic differences in both this resolution, because now you can see individual cells, but also in this detection efficiency from this direct in situ detection. So here, for example, with 55 micron grid spots, you can't identify what an individual cell is doing, but rather you get the expression averaged over all of the cells within that grid spot. But also with Merfish, with the direct in situ detection, we detect about 70-fold more transcripts than with previous oligo array-based technologies. So with that, you can start to understand what these moderately or moderately lowly expressed genes are doing across the tissue, such as this opioid receptor delta 1, where within this region of the brain, you find it's very highly enriched within specific cell types within this region of the brain. But beyond the transcriptomics, there's also the other unique aspect of this spatial profiling technology is that it's entirely compatible with proteomics measurements. So in fact, just a month or two ago, within Vizgen, building upon the platform that we released earlier this year and entirely compatible with that, Merscope platform is the ability to measure proteins simultaneously with RNA. So currently we do that through antibody secondary labeling approach, so indirect immunofluorescence. And we have an oligo attached to that secondary antibody, and Vizgen can provide these oligo labeled secondary antibodies. So then with that, you're able to combine the secondary with your primary of your choice. And in this way, simultaneously measure 500 genes plus 5 or 6 proteins within the same sample. So you can start to see how the protein expression correlates with the RNA expression, whether you're trying to use the protein to identify certain structures, even at a subcellular level, whether you could stain it, for example, to mark the ER and look at which transcripts are co-localized with the ER, or any other structure component, either within a cell or across a whole tissue. You can also use that protein to help identify certain cell types, for example, or see how well the RNA corresponds with the protein in certain cases, because maybe sometimes the difference between the protein and the RNA could be biologically informative. So we're very excited to, on top of the RNA capabilities that have been in the platform since we started shipping in summer of 2021, to now have this custom protein compatibility where now you can do a multi-omic measurement with the Merscope platform. But to understand Merfish technology best, sometimes it's easiest to get your hands on the data. So within Vizgen, we have released a number of public data sets from mouse brain to mouse liver to a number of human FFPE oncology data sets that are all publicly available on our website. So if you are interested in seeing exactly how Merfish and Merscope data looks and start to think about how it might apply to your research, feel free to check out these publicly available data sets, because they're very similar to the data that you could get out of the Merscope platform. So from Merfish technology, which, as I mentioned, was originally published in 2015, Vizgen's focused a lot on putting together this commercial solution for Merfish technology that starts to enable a rapidly growing number of labs to start to use the technology for their own research. So in this next section, I'll discuss how does the Merscope platform look when you are going through the process for Merscope to take your sample and get to the biological insight from that sample. So the centerpiece, of course, for the Merscope platform is the Merscope instrument. But building on Merfish technology, we wanted to maintain all of the advantages of Merfish technology, this high sensitivity, this multiplexing capacity, and this accuracy, with easy to use tools to make it easier for any lab that's to be able to focus on the biology rather than the technical details of the measurement. So since we offer entirely custom gene panels, the first step in that process is to design your custom gene panels, to specify which genes are most relevant for the biological question that you're working to address. So to facilitate that, we have this web application where you're able to upload the list of genes that you'd like to target, and then it gives immediate feedback about whether those genes are suitable for a Merfish measurement, both based on the length of the transcript, but also the abundance. So with that feedback, then you can hone down the list, move the highly expressed genes into a sequential panel, maybe add additional genes if there's additional need, and come up with your final gene list. Once you have that gene list specified, you can place an order with Vizgen, and then Vizgen provides that gene panel in a ready-to-use aliquot. So then you just take your tissue block. You'd want to make sure that it has good quality RNA in the first place, since with any transcriptomic measurement, the quality of the RNA does impact the quality of the output that you get from the platform. But once you verify that that tissue block has high quality RNA, then you can slice it, fix it, permeabilize it, and stain it with this gene panel. So this is the set of oligo probes that bind to each of the different targeted transcripts and encode this binary barcode onto each of those transcripts. So there's a first stage of hybridization that happens on the bench. Then after a gel embedding and clearing process, which clears away the autofluorescence background across the tissue so that we can have sample compatibility across very wide range of different tissues, then you're able to load it into the Merscope system. So the Merscope slide goes into the flow chamber, and then along with the reagent cartridge, they get loaded into the Merscope system. Within the system, we have a 2 square centimeter imageable area. So it does a quick low magnification scan of that 2 square centimeters of tissue. From that low magnification scan, you can select your areas of interest. Where did you actually place the tissue within that larger imageable area? And then from there, once you have it set up, the instrument can proceed automatically to go through the sequential imaging process in order to identify the barcodes at each position across the tissue. Once it finishes that process, it takes all of the raw images that it acquired during the sequential imaging and within the system decodes them in order to create the more biologically meaningful outputs. So it decodes the images to create this list of detected transcripts. The list of detected transcripts contains XYZ coordinates and the gene identity for each of the transcripts that was identified across the sample. It also, out of the box, performs cell segmentation. So it provides a cell by gene matrix that you can automatically, or with little effort, put into any tool that you're familiar with for single cell sequencing, whether it's SCAMPI or Seurat, to cluster that cell by gene matrix and identify the different cell types across your tissue. We also, since some of these files become quite large and it's not always trivial to plot out hundreds of millions of points, even directly through Python, we have built out interactive visualization software that lets you easily toggle through all of the genes within your measurement and see how they're expressed across the tissue slice. So just to summarize, the Merscope solution contains tools to facilitate everything from custom gene panel design. Currently, we offer entirely custom panels of up to 500 genes. As you see with the protein kit launch, we're also working on additional features on the platform to support even larger gene panels, all on the same hardware. From that, once you have your gene panel in hand, there's some benchtop work to stain the sample. It's maybe 3 hours of hands-on time, a few overnight incubations, but that gets the sample ready to go onto the instrument. Then it goes onto the instrument. It takes about a day per sample of imaging time within the instrument, and then about a day of processing time. And then you get these output files, your list of transcripts, your mosaic TIFF images, your cell by gene matrix, your cell metadata, which is the spatial information about each of the cells, and the full cell boundary polygons. So there are more detailed slides, but maybe we can come back to them if there are additional questions, because I do want to make sure we have time to discuss both some of the Merscope applications, but then also the FFPE compatibility, which we officially launched just last week, and we're excited to have now Merscope compatible with both fresh frozen, fixed frozen, and FFPE tissues. But as a genomics tool, it really applies to a wide variety of different applications, because any biological system, if there's more than just one cell within that biological system, there is some spatial arrangement, as likely some cell-cell communication or interactions between those cells. This includes anywhere from neurodegeneration, where understanding which areas of the brain are impacted by the neurodegeneration and understanding how the gene expression is changing within that vicinity versus a healthy brain, and exactly which cell types within the vicinity that's happening in, gives additional insight into how these different neurodegenerative diseases are progressing. So that's one very spatially arranged, broad application area. Similarly, with cancer immunotherapy, understanding where the immune cells are across the tissue, how far they're able to infiltrate into the tumor, what their state looks like in different areas of the tissue, gives insight into how does the microenvironment of the cancer impact the ability of the immune system, or whether it's the host's natural immune system or whether it's introduced immune cells to help fight the disease. And along with that, any infectious disease naturally also has rich spatial organization, because there might be certain cells across the host tissue that are infected either next to a bacteria or infected with a virus, and understanding how both that cell that's infected is impacted by the virus, but also how that's communicating or interacting with the surrounding cells gives more insight into the progression of those different viral or bacterial diseases. But this is just a small example of the different applications that we're already seeing Merscope applied to so far. To help towards that end, to help researchers just focus on the biology rather than the technical details, within Vizgen we have demonstrated Merfish on a whole variety of different mouse and human, and even beyond that, non-human primate, rat, even some plant tissues, where we've had, as it's a fish-based technology, it's been fairly straightforward to apply it to this whole range of different tissue types. And with that, it opens up this broad avenue of biological research. So the first example of Merfish technology goes back to even before Vizgen was started. One of the initial key biological demonstrations of Merfish, which is this 2018 science paper from Xiaowei Zhuang's lab, where they demonstrated, as they were developing Merfish technology, they demonstrated the ability of Merfish to very accurately profile cells across the mouse hypothalamus. So here, this is a region of the brain known for different aggression, mating, and other sorts of behaviors, and they wanted to build up this molecular and cellular, and as we'll see on the next slide, even functional atlas of this region of the brain to better understand how these different subtypes of inhibitory and excitatory neurons that they see could be implicated in these different behaviors. So since Merfish was at the time still a relatively nascent technology, they did this in very close conjunction with single-cell RNA sequencing, just as a way of validating the results, validating that what they see with Merfish is consistent with what people are familiar with from other technologies. So with Merfish, they measured about 500,000 cells in the control case, about 1.5 million cells in all. But from the t-SNE, you can see this very rich subclustering of the major cell types. So here, this t-SNE is colored based on the major cell types across the brain. But you can see within those major cell types, there's many subclusters that they were able to pull out. So when they look at the major cell type level, they do find very good correspondence between the Merfish measurement and the single cell RNA sequencing measurement. But when they go to the next levels, or subtypes of inhibitory and excitatory neurons, when they compare that to single cell RNA sequencing, again, they find very good correspondence where almost all of the clusters they identified with Merfish are also present in the single cell RNA sequencing clustering analysis. But there are even some subtypes of inhibitory neurons where they found they were able to more finely differentiate the cell type than even with single cell RNA sequencing data. So those are in this plot on the bottom left. Those are the two clusters towards the right hand side, where there's a single cell RNA sequencing cluster that corresponds with two different Merfish clusters. So here, when they looked into this more, they did find that those were in fact true cell types based on additional fluorescence in situ hybridization measurements, but they also found that more fine differentiation of Merfish was driven by the higher sensitivity of Merfish. In particular, those subtypes were expressing different neuropeptides, which are very functionally relevant in this region of the brain, but also lowly expressed, so dropping out of the single cell RNA sequencing measurement, but accurately detected with the Merfish measurement. So with that, using just this panel of 155 genes, in this case, they were able to build out this transcriptional and cellular map of this region of the brain, discover novel cell types that were not even identified with single cell RNA sequencing, and create this map of the MPOA region of the brain. But on top of the transcriptomics measurement, they wanted to understand which of these subtypes of inhibitory and excitatory neurons might be implicated in the different types of behaviors that this region of the brain is known for, the parenting behaviors, aggression behaviors, and mating behaviors. So here they perform Merfish in combination with measuring an early activity reporter gene, CFOS. So then they were able to look at which specific subtypes of excitatory and inhibitory neurons across the brain were activated during these different behaviors. So then they could build up this functional map of the MPO region of the brain in ways that would be impossible with preceding technologies. So with this, by functionally mapping which neurons were activated with during certain behaviors, gives them more insight into what is the biological role for all of these different excitatory and inhibitory neuron cell types that they're seeing across the brain. A couple other applications that I want to point out briefly are some that go beyond the brain into, for example, mouse kidney, and rapidly growing number of different other applications. I think on Twitter, I just saw some lung data coming out of UCSD that seems very exciting. But beyond brain, there's quite a bit of other very intricately organized tissues. Here from WashU with Ben Humphrey's lab, they were looking at kidney profiling using this panel of 243 genes, in particular looking at sexual dimorphism and kidney damage between males and females, where the females seem to be able to recover from the kidney damage much quicker. So they wanted to use Merfish and Merscope to better identify what are the molecular mechanisms for that faster recovery, which could have downstream implications for better understanding the kidney and better understanding the kidney response to these types of damages. But just last week, we did also release our FFPE kit. While for many biological questions, there is fresh frozen tissue that's readily available, there's also a broad range of different questions that could come up where FFP might be the only type of tissue available, whether it's because the FFP has been banked along with a clinical trial, and then you want to go back retrospectively, look at what do those samples look like. Maybe it's a sample that contains a hazardous pathogen where the only way that you can safely handle it is if it's heavily fixed to begin with. But these also have certain challenges because the RNA could start to degrade. So in some cases, there could be optimizations to the protocol that we found were helpful to adapt it for the FFP tissue type. So when we were creating the FFP protocol, we did want to make sure that it was very consistent with the fresh frozen tissue protocol. So the same quality of data could come out at the end. So here with the FFP workflow that we've optimized, you get very good correlation with bulk sequencing, as we saw, very good reproducibility between technical replicates, and very good correlation between the fresh frozen and FFP protocols. So in fact, when we look at as method development types of samples, when we look at fresh frozen mouse brain and FFP prepared mouse brain, and we do Merfish measurements on each using the same gene panel, then when we cluster them, the clustering doesn't separate based on whether it's fresh frozen or FFP, but rather it separates as you hope based on the cell type. So here is a UMAP containing both the fresh frozen and FFP samples. And when you see it compared to, so we can turn on the animation. Right, and then this will animate to the spatial coordinates. So you can see that as it animates, these different layers across the brain are very consistent between the FFP and fresh frozen, where because these two separate protocols are so similar, you are able to pull out the biologically significant transcriptional differences rather than any transcriptional difference that could be due to the way that the sample was prepared. With this FFP, as I was mentioning earlier, we also have an FFPE data release that I'd invite all of you to look into in more detail. But here we have almost a square centimeter of FFP ovarian cancer. Just as with the brain, you can look at the transcriptional profile across the whole tissue. But again, zoom in to the cellular environment, the cellular neighborhood, all the way down to the subcellular organization. So here we're mapping this panel of 500 genes across this full tissue type, accurately identifying the major types of cells and even subtypes of those major types of cells in order to characterize the spatial organization of this tissue. With the FFP, then it opens up a broad range of different sample types, in particular human sample types, where FFP could be the only way that some of these samples could be accessed. So anywhere from human ovarian cancer to human lymph node to human tonsil, human skin, and human kidney. So we're very excited to have released that FFPE protocol last week and excited to see the sorts of applications that it will start to enable. But as with the protein kit that came earlier, within Vizgen, we are very much dedicated to continuing pushing new functionality onto the platform, just as we did with this protein and FFP kits. And we have a lot of exciting new functionality coming on the platform in 2023. So with that, we could likely use the last few minutes to address questions that came up. But if you are interested in the Merscope platform and how it might apply to your biological research, feel free to get in touch with me or the Vizgen team with additional questions.
Great. Thanks so much, Dr. Emanual. So if folks do have questions, feel free to add them in the chat or raise your hand. We can take them. Those are really great overview of the capabilities of the Vizgen technologies and also sort of like the logistics of how single cell spatial is working. There's a question from Jennifer. So with the FFPE compatibility, is a digitized H&E image generated along with the data? Looks like there's a series of questions. Maybe I'll just start with that one.
Yeah, I can read through them as I go along.
Sure.
Well, but for this first question from Jennifer, in terms of H&D, so with each measurement, we do include a stain for the nuclei. So it's a DAPI label of the nuclei. So there are ways that you can simulate a type of H&D. We don't have out-of-the-box solutions for those, but we do have these additional markers that help identify tissue morphology. So that's DAPI. We have a stain for poly-T, which is the total RNA content. And then for these dense tissues, we also have a cell boundary staining kit, which we recommend for cell segmentation in these dense tissues because it marks the boundary around each of the cells. So these stains can be used to understand the tissue morphology, but it's not a direct out-of-the-box H&D analog. For that, there are different solutions that could be used. For example, adjacent slice, putting it through H&D. There could also be opportunities to do H&D first and then go through the Merfish protocol. But so if something along the lines of exactly similar to H&D is required, there are options, but there is a lot of tissue morphology information in the stains that we already image and the images that we already output. So then following that question from Jennifer, there's one asking about the fixation time and age of FFP blocks and how that affects the assay. So since Merfish is a technology where we are profiling the RNA, within the tissue, the quality of the RNA does impact the quality of the measurement. So it's not so much based on necessarily fixation time or age of the FFP block, but even some blocks that we've seen stored for quite some time could still have RNA within them, intact RNA, and we still can get good measurements from them. On the other hand, there could be tissue blocks that were acquired very recently where maybe there was RNA contamination of the tissue and no RNA left, where there might be more challenging to get high quality measurements from that tissue. So here, instead of age or fixation time, we do have guidance on characterizing the RNA integrity with DB200, where we'd recommend DB200 above 60, but can still produce good quality measurements from DB200 less than 60, but would recommend, especially if it's the first samples that you're running, to pre-screen them for the RNA quality. Yeah, so that gets into the next question as well. What is the recommendation for testing FFP tissues for quality? So there we always recommend the DB200 via tape station or some other instrument, but we also have a verification kit. So we have a reagent where you're able to stain a single gene through the same protocol that you use for Merfish, and then see how bright those spots show up in the end result. So it gives you a much less expensive way of evaluating tissue quality than a full Merfish measurement, and helps you maybe screen out the tissue blocks that might give the best quality measurement. The next question is about point mutations and whether we can detect point mutations when we design the probes. So currently with our probe design software out of the box, we don't have built-in capabilities for point mutations. It is something we consider if there is a particular use case that you're interested in. I'd be happy to discuss more, but currently out of the box with Merscope, we don't have assay for detecting point mutations. Next question is, do you use specific genes to perform cell and nuclei segmentation? So here, rather than the transcripts, we tend to use these additional stains like DAPI or the cell boundary stain to draw the boundary around each of the cells. So there, DAPI can help mark the cell nucleus, and then the cell boundary can help mark the total cell area. So we don't use specific genes, but we do use these specific stains to help out with the cell segmentation. Then the next one is, how much does it cost compared with single cell RNA sequencing? So here, there's different ways of comparing the cost. Maybe the most straightforward one is sort of per sample. So for a single run on the Merscope instrument, which could contain multiple tissue sections, but the reagents are effectively priced per Merscope slide that goes through the full workflow, and that comes out to about $3,500 all in for the custom gene panel, the sample prep reagents, and the imaging reagents for that sample. So within that measurement, then you can get, in some cases, 500,000 or more cells. So if you compare a per cell cost to single cell RNA sequencing, that becomes much less expensive per cell than single cell RNA sequencing. Often with single cell RNA sequencing, as you go through the dissociation process and the barcoding process, there's certain cells that might drop out. But here with the image-based approach, you're in essence capturing every cell across the tissue. Then the next question is for subcellular resolution. So we do have much better than cell level resolution, where it is single molecule resolution. And we know the location of each of these individual transcripts that we detect to well within 100 nanometers of its position within the biological tissue. So there, you do have much better than single cell resolution. And then with that subcellular resolution, you could, for example, look at co-localization between a transcript and a protein stain with that very high resolution. So all the output on all the Merscope measurements is taken with this very high resolution. So then the next question is, what is the cost comparison be between Merfish and other spatial platforms such as Vizgen? So with that cost comparison, one of the maybe key pieces is that with Merscope, there's no need for sequencing. So with Vizgen, for example, you have to purchase the base slide from one company, and then you have to purchase the sequencing from another company. So in some sense, each one, it's almost paying twice for the same data. With Merscope, through the same workflow, you can get to the end biological results without any need to put anything through the sequencer. So in that sense, I probably this per sample cost is most intuitive. It's not perfectly one-to-one because you have the square centimeter of tissue area with the Merscope measurement, where with Vizgen, it's the smaller grids, but you can always put smaller tissues within the Merscope slide arrayed as a TMA or some other tissue array and get multiple tissue slices within the same Merscope within a single Merscope sample. So I don't have an exact number compared with Vizgen, but there are certainly these technical advantages to having the single molecule and single cell resolution.
And I think Roman had a question about the Merscope instrument on our campus. Yeah, we recently do have one on campus. It's located in somebody's lab. And so if you're interested, you can reach out to us at SCSAP and we can try to coordinate access. It's not located in a core facility currently. And then I think Roman's other question about how stable are the Merfish probes once they have been made is also a relevant one.
Yeah, that's a good one. Currently with the Merfish probes, we provide them in aliquots of for 20 samples, and that has a shelf life of about 4 months. We are working to extend that shelf life for the Merfish probes, but we also are working on pre-designed panels where maybe for your first measurement, you don't necessarily need to come up with this entirely custom gene list if you just want to see how Merfish technology works on your sample. So there with those pre-designed panels, we're also going to provide them in smaller aliquots. So you can buy just enough for 4 samples or 12 samples, for example.
Great, thanks. And then one other question that I missed because I have a lot going on in my chat. So Wei Shang asked, is it possible to get subcellular resolution?
Yeah, there it came through it as I was reading through the questions, but that we have the single molecule resolution, so much better than the cell scale resolution, where you know exact position of each transcript to within 100 nanometers or so.
That's excellent. Thanks so much. And then it doesn't look like there's any other questions. Oh, here we go. So yeah, so Roman had a question that I had as we're like thinking about like more complicated cell types and cell shapes. Can you detect transcripts and axons?
Yeah, so in this brain section, I might be able to go back to it fairly quickly, but you see that at least in a tissue slice where we are taking this 10 micron thick slice of tissue. Let's see, is that coming up? So if we go into where it's zoomed in a bit, let's see, right. So when it's zoomed in, you can see that there's these transcript dense areas of the tissue. And within those areas, it corresponds with the cell body or the soma of these different neurons. But you can also see that there's transcripts that are within those cell bodies, or sorry, outside of those cell bodies where we're still identifying transcripts. So those are likely transcripts that are within axon or dendrite. But with a 10 micron thick tissue slice, it's hard to trace an axon all the way back to a given cell body because that axon likely goes outside of the 10 micron thick tissue slice. So you're still detecting the transcripts within the axon or dendrite, but assigning them back to a cell body is somewhat more challenging in tissue. On the other hand, in cultured neuron systems, we have shown that since now those cultured neurons are growing along the glass surface, it's very easy now to trace the axon or dendrite all the way back to the cell body. So there is a publication coming from Zhao Wei's lab from a year or two ago where they did that, where they looked at how different transcripts were enriched along different lengths of the accent or dendrite, so map out the transcriptional profile. So there's possibilities easiest in cultured cells, but there's likely still analysis you could do with these transcripts in the tissue, even though you might not be able to tie them specifically to a given cell body.
That's great. And so maybe let's do one last question, Tom.
Yeah, I don't have a question. I just wanted to give a little bit more information on the Merscope that's on campus. So it is in a research laboratory in the LSI, but the Advanced Genomics Core and the Spatial Analysis Program we are working on an arrangement to be able to gain access to that. So for example, through the Advanced Genomics Core as part of our spatial offerings, really what we need to proceed is a project and a probe set. So the fact is we've been ready for a few weeks now to try to see where we can go with this, but we really need a project, and maybe that'll lead into the next thing we're going to hear about. So
great. Thanks for the context there. I appreciate it. That's helpful. Yeah, teacher, do you want to take it away?
Yeah, so we're finalizing the details. So first off, thank you for everybody for joining us today. It's the last couple minutes, so I don't want to take up too much of your time, but really, we will be having a grant project for the researchers at University of Michigan. We're finalizing the details. Vanessa will be sending out a link to it, hopefully very soon. Legal is always fun to deal with at any institution and within a company. So the general gist of it will be that we will we will work with you to design a human or a human immuno-oncology panel, right? It will be a 500 gene set. We will have the final probe set available when the grant is available. It'll be for human FFPE samples. It will be enough for 2 experiments. So if you want to do TMAs, et cetera, that is perfectly fine, as long as, it fits on 2 different slides. That's what it comes down to. It'll all be free. That's the fun part about a grant, right? Everything will be cost will be covered by Vizgen. We will give you discounts for for that project for future reagents needed as well, so that you can, if it's for a publication, et cetera, you can complete it. But most of the time, 2 slides will be more than enough for what the project is. We're excited to work with everybody on doing this. We will, as I said, hopefully have details relatively soon about this.
Great. Thanks so much. Yeah. And as those details come up, we will make sure to share them with our community. So I don't see any other questions. With that, I'd like to thank our guests so much for joining us and telling us all about the exciting technologies coming out of Vizgen and these great opportunities coming up. Vanessa, any closing thoughts, parting words?
Yeah, thank you so much to our guest for coming and presenting, and for everybody else on the call and other membership that I'll be emailing. We will see you in 2023, and happy holidays to everybody. Thank you so much for spending your time with us.
Happy holidays, everybody.
Great, thank you.